Welcome to Botany class, Chapter 2, Mineral Nutrition, Part 1. In this, we will discuss about methods to study the mineral requirements of plants. In 1860, Julius von Sachs, prominent German botanist, demonstrated for the first time the plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique it is called as hydroponics. The technique of growing plants in a specified nutrient solution is known as hydroponics. Hydroponic plant production. Plants are grown in a tube or a tube placed on a slight incline. A pump circulates a nutrient solution from a reservoir to the elevated end of the tube. The solution flows down the tube and returns to the reservoir due to gravity. EAMCET shows a plant whose roots are continuously bathed in aerated nutrient solution. The arrows indicate the direction of a flow. The minerals present in the soil can enter plants through roots. In fact, more than 60 elements are found in different plants. Some plant species accumulate selenium and gold, and some accumulate radioactive strontium. Criteria for essentiality. In this, element must be absolutely necessary for supporting normal growth. Requirement of the element must be specific and not replaceable. Element must be directly involved in the metabolism of the plant. Based on this, elements are further divided into two broad categories based on their quantitative requirements. One is macronutrients and one is micronutrients. Macronutrients are generally present in a plant tissue in large amounts, excess of 10 mol kg dry matter. They include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and magnesium. Here, micronutrients are trace elements are needed in very small amounts, less than 10 mol kg of dry matter. This include iron, manganese, copper, molybdenum, zinc, boron and chlorine and nickel. Essential elements can also be grouped into four broad categories. One is elements that are components of biomolecules like carbon, hydrogen, oxygen and nitrogen. Second, essential elements that are components of energy related chemical compounds in plants. Example, magnesium, chlorophyll, phosphorus in ATP. Third, elements that are active or inhibit enzymes. Example, Mg2+ is an activator for both ribulose phosphate carboxylase. Fourth one, essential elements can alter the osmotic potential of a cell. Here, example, potassium plays an important role in the opening and the closing of stomata. Another topic, role of macro and micronutrients. First, we will discuss nutrients are essential elements which act as a macro elements. First one, nitrogen. It is an essential mineral nutrient required plants in the greatest amount in the form of NO3 minus NO2 minus or NH plus 4. Here metabolically the tissue active from nitrogen and constitutes of proteins, nucleic acids, enzymes, vitamins and hormones. Second is the phosphorus. This phosphorus absorbed by the soil. So these ions which are phosphate ions H2PO4 minus, HPO4 minus. Here the phosphorus which is present in proteins, all nucleic acids and nucleotides. Potassium is absorbed as a potassium ion K+, required in most abundant quantities in the tissues, buds, leaves and root tips. It also helps in opening and closing of stomata and activation of enzymes. Calcium. It is also absorbed from the soil. Calcium ions Ca2+. It required meristematic and differentiating tissues during cell division. Synthesis of cell wall. Calcium pectate in the middle lamella and it accumulates older leaves, and involved in the normal function of the cell membranes. It also plays a major roles in enzymes and regulating metabolic activities. Magnesium. It is absorbed by the plants in the form of divalent magnesium ions and activate enzymes, photosynthesis, and synthesis of DNA and RNA. Magnesium is a constant of ring structure chlorophyll and maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate, SO2 minus ions. Sulfur is present in two amino acids, cysteine and methionine. Here, these are also present coenzyme vitamins, thiamine, biotin, and coenzyme A, pyridoxine. Sulfur forms disulfide bridges, help stabilizing the protein structure. Then iron. Plant obtain iron in the form of ferric ions. Iron is required in large amounts compared to the micronutrients. Here the pyridoxine and the cytochromes which are involved in electron transfer. So reversibly oxidized Fe2 plus Fe3 plus during electron transfer, and it is activates the catalyze enzyme. Next, manganese. It is absorbed from the manganese ion Mn2 plus activate enzyme in photosynthesis, respiration and nitrogen metabolism. Manganese in the splitting of water to liberate oxygen during photosynthesis. This is very important in a NEET. Zinc. Plants obtain zinc Zn plus 2 ions, activate various enzymes, especially carboxylase, and synthesis of oxide. Copper, cupric ions essential for overall metabolism in plants like iron, Ca2 plus and Cu plus. It is also a structural component of electron carriers like cytochrome C oxidase, and mitochondria, plastocyanin in chloroplast. Then boron. It is absorbed as BO3 minus or B4O2 minus ions. Boron is required for the uptake and utilization of Ca2 plus and elongation of a cell, differentiation and carbohydrate translocation and pollen germination. Molybdenum. It is also obtained form of molybdate MoO2 plus and a component of several enzymes including nitrogenase, nitrate reductase, participate in a nitrogen metabolism. Chlorine. It is absorbed in the form of chloride ion Cl plus, Na plus, Clay plus. It helps determining the solute concentration, anion cation balance, and essential for the water splitting reaction in a photosynthesis and oxygen evolution. Nickel. It is recently recognized as a 17th essential nutrient as it acts an activator for urease, important enzyme of nitrogen metabolism. It is also found in induced disease resistance in some plants. So this is all about micronutrients and macronutrients, essential elements.